Hello everyone, and welcome to the SurvivalG tool. To start, please provide your email address and name of organization, then hit the Submit button. This will bring you to the web interface with two input panels. There's a panel on the left and a panel at the top. A brief introduction is provided under the About tab. A step-by-step guide is also included here under the Manual tab. This provides users with details on how to navigate this tool, including options available to users and how to use this tool. You may want to read through the entirety of the manual before you begin your analysis, so you understand all the different options SurvivalG has to offer. In addition, the manual also walks through example uses of SurvivalG. In order to begin the survival analysis, the users first select the type of analysis to perform. The analysis options include a gene-based analysis, which is to query single gene expression or gene set enrichment. A cell-based analysis, which is to query immune cell composition. This is based on estimation of tumor infiltrating lymphocytes and bulk tumors expression data or H&E imaging data. A profile-based analysis, which is to query weighted gene co-expression. The weightings for each gene in the signature is provided by the user in the input data file. And finally, a mutation-based analysis. This is used to query the effect of tumor mutation burden, or TMB, within a cancer type by the total number of non-synonymous somatic mutations, exonic mutations, and all somatic mutations per MB. When users opt for gene-based analysis, they can choose from the four following options. First, you can choose cluster markers from a single-cell RNA-seq dataset. Second, they can input a gene set, which is a set of genes that can be provided as a list from a pathway or a biological process. Third, they can input an expression ratio of two genes to study their associations with survival outcomes. And finally, they can just input a single gene of interest. Now, we are going to show an example use of SurvivalG. To start, we are going to use the gene-based analysis tool, specifically the single gene option. First, the user will input their gene of interest. We are going to use FLT3. Next, the user will click the Check My Gene button. If the gene is contained in the dataset, then the user can continue with the analysis. Next, the user chooses a cancer program from those provided here. They can click the question mark to show more information about each program. Then, the user selects a dataset from those provided by the cancer program. We are going to choose the TCGA LAML dataset. Under the Select Tumor tab, the user picks the sample tumor type of interest for their analysis. Under the Select Groups tab, the user chooses how they want SurvivalG to split the samples into high and low expression groups. The user can choose from overall or event-free survival, and finally, the TAL tab is used when the user chooses cell-based analysis option. The user clicks Run, SurvivalG performs the analysis with the parameters selected. Here are all the output plots for our FLT3 gene-based survival example. Now we are going to show an example use of the gene set option of SurvivalG. This is similar to the single gene option, except the user can upload their own list of genes or choose from one of the examples provided. We are going to input a list of 7 example genes and then click Check My List to make sure they are contained in the dataset. This time, we are going to choose the target cancer program, and we are going to select the target AML dataset. Next, under the Select Groups tab, we will choose the Cut P option. More details on this group selection method can be found by clicking the question mark of the Select Groups tab. Again, we click Run, and we wait for SurvivalG to produce the results for our input parameters. And here are the results for the gene set analysis. In this example, we see this gene set has a high hazard ratio in the target AML dataset. And finally, thank you for using SurvivalG.